So we're going to go ahead and get started with our first speaker. We are going to learn about applying the principles of the science of safety to improve subject safety in clinical research, actionable practices to identify and minimize risk. Allow me to introduce to you Dr. John Kessler.
Thank you very much. And I think to set the tone, there's a tone. I get an extra 1 minute now. Okay, excellent. So to set the tone for the content of my talk, I thought I would give you some context about me. I have been in pharmacy for 45 years now. I have been direct clinical practice and direct patient care. I've worked in informatics, clinical research. For 15 years, I ran a research pharmacy at an academic institution you may have heard of called Duke University. And I wore a suit as part of my career. So all of that composite is what makes me who I am today professionally, and why I'm here to speak to you today is about safety. Safety became a really important part of my clinical practice and in my research interest over the years. And what I want to propose to you is that there is a lot to be learned from clinical practice safety that can be generalized to clinical research. So these are the things that we'll talk about today. They're in your handouts. I won't read through the details because time is short. But to begin with, clinical research, the facts, for sure, you in science are producing amazing discoveries through the dedicated efforts that you put into your work and the efforts of your colleagues. There's no doubt about that. That's the premise of the good things that we do. The rest of the talk, not quite so pleasant. So I'll try to smile as I talk about things that go wrong that we want to talk about. Clinical research has a perception that it's highly scientifically controlled, that it's carefully conducted. Clinical research is done under precise execution of protocols by highly qualified study teams. And on top of that, our research is overseen and regulated by the FDA and IRBs, again, to assure subject safety. Again, those are the perceptions and in part the reality of where we are today with research. However, let's just take a look at one of those in a little detail. Take a look at the IRB, okay? Because having been on an IRB for 27 years, having been the chair of an IRB for 11 years, these are things that I've noticed. So observations to me are that the IRB doesn't see, from a safety point of view, anything about the drug, its packaging or its labeling. They don't see the dosing and calculation worksheets that are used to determine the dose of the IP. We don't see information about how the clinical staff in an organization work hand in hand with the research staff in an organization from a roles and responsibilities point of view to know who's caring for that subject at any given point in time. All of these things are silent to the IRB submission. Each of these things are important to subject safety when you think about the design of how research interfaces with clinical practice. And because of that, sometimes things go wrong. So the premise of the talk today is that those of you in clinical practice or who have been in clinical practice know that there has been, for more than 20 years, a national patient safety movement that's made important advances in reducing patient harm from medical care and medication use by three principles: learning from mistakes, developing a culture of safety, and instilling that through the organization at all levels, from leadership through the front line, and then developing systems that not only analyze harm, but as importantly, analyze risk and near misses. Potential harm. In contrast, I'll propose to you, perhaps even debate with you over a nice scotch, that there is no national patient safety movement equivalent to that in clinical research. There is no movement to reduce the unsafe practices that lead to preventable harm by unsafe research, design, flawed infrastructures, leadership failures, and lapses or absences in the culture of safety in the organization. That's the premise. So let's look at that. Well, in clinical practice, we talk about a patient safety movement that is building upon a new science of safety. And in clinical practice, what that means is it's a new way to look at the way that care is delivered. And I'm going to say that we need to say a new way to look at the way that research is delivered. And we look at that through our understanding of the complex systems that affect subject outcomes. We understand the errors, and we call them in research deviations, are actually rooted in flawed systems design and not individual performance problems. The new science of safety says that individuals are rarely the root cause of the problems that occur in research, and that for all of this to work well, there has to be a culture of safety critical to the positive outcomes. And just as a sidebar, the science of safety concept in clinical practice that I'd like to see moving into clinical research is different than the FDA's definition of the science of safety, which focuses on preventing drugs from causing adverse events through pharmacology and other mechanisms. So there's a little difference, and I want to build that contrast as we move through here. So what I've done is to help show the points that I'm talking about. I thought, well, one of the lessons in patient care is to learn from mistakes. Well, let's learn from these errors and deviations that occur in clinical research. So if you wanted to find a national database of medication errors in research, you'll not find that. There is no national database of the problems with medication use that goes wrong in research. For that matter, devices, nor any other aspects of care. There simply is no organization of what's happening in practice. So what I did was to say, well, maybe one thing I can do is look at the unanticipated problem reports submitted to the OHRP as one measure of the kind of things that go wrong in research, at least those that reach the level of being unanticipated problems. And in 2009 through 2010, over this 3-month period, we submitted a Freedom of Information request to OHRP to gather 85 unique case reports involving 150 subjects, is what the results of the FOI returned to us. I submitted the same request to the FDA and was denied access to any of the records from the FDA, as these were considered proprietary information under an IND and therefore not released for research and analysis purposes. But OHRP complied with the request. Not that they were happy, nor did they have a database. They had to go through every file to find the reports of unanticipated problems, and they sent me a big CD full of these. 17 of these involved medication errors from 10 different U.S. institutions over a 3-month period of time. And what we did was to do a retrospective analysis of the type of problems and the contributing factors, the corrective actions, and other demographics of that data set to try to learn what's happening in research. So what did we find? Well, we found a number of medication errors. In the wrong drug error category, and these were medication errors where the drug was prepared and administered, we found that cyclophosphamide was given to cancer patients instead of cytarabine. What was the corrective action to that? Amazingly, that organization conducted a root cause analysis. That's the right action when you have an event that serious. And following that root cause analysis, now remember, this is like in 2009, they had 2 nurses do a double check of the chemotherapy prior to administration. That's great. But that contrasts with clinical practice, which had been done double checks on medication administration of these kinds of drugs for 2 decades before that. lesson. RCAs are great. There were safety practices that we knew about from clinical practice that were not being translated into clinical research. The second one is an example of what we'll call a look-alike drug. These were two compounds labeled exactly as you've seen them on the slide that were mixed up in a clinical trial. The contributing factors weren't specifically identified, but you can guess by the corrective actions that they were stored together on the same shelf. Now they're physically separated. They have special stickers to help distinguish the look-alike drug names. And then the organization added, again, a double check of the product. They also took retraining and other personnel actions, perhaps punitive, hard to know from the report, but nonetheless, this is a at-risk drug nomenclature because of the names that look alike. There were wrong dose errors. These are a sample of the wrong dose errors that came out of the trial, with 40% Taxol overdoses, a chemotherapy drug. There were underdoses of Mesna, which is dangerous because it's protective to the bladder when you're giving cyclophosphamide to prevent cystitis. So those patients were at risk of cystitis because of the underdose. There was a twofold cisplatin overdose. There were underdoses of growth hormone injection, overdoses of other chemotherapy. All of these things happened, but to me, the striking point and the lesson learned here is that some of these happened for 22 consecutive days. First day you miss it, and then it was missed 21 more days after that. So where were the checks and balances in the system to see that the dose was correct every day? The MDX, 2 patients overdosed for 3 days each, and again, the growth hormone happened for 14 days. So the message is, once a problem starts, it will continue until somebody intervenes and recognizes that that problem is occurring. And that requires systems of safety, a culture of safety, and it requires the vigilance that things can go wrong. So the contributing factors related to wrong dose errors, as best as we can discern from the OHRP reports, included inadequate verification of the dose when it was specified. It included failures in the verbal order process, giving verbal orders for investigational drugs. Bad idea. Not a great idea in clinical practice, not a great idea in research practice as well. What did they do? Education. They educated people. Education seems to be the tool the remedy in our toolbox to fix problems. Education alone is at the lowest level of effectiveness on the hierarchy of correctable actions following problems. So things that go wrong, it's not so much what went wrong, but it's understanding the contributing factors and the causes underlying that, and then developing corrective actions that align with the contributing factors. Additional examples here, there was an ad hoc change in an IV concentration requested by a nurse, and the pharmacist preparing the medication wasn't expecting that ad hoc change in the concentration, and there was a mixing error in that particular product. Transcription errors, and then doses of drugs based on inappropriate interpretation of other diagnostic signs. And so you can see that what was important here is to understand why it happened and then align the corrective action to those underlying failures. There were examples of clinical trials in which patients were exposed to an entirely different drug, and in this case, 67 different subjects at multiple sites received lidocaine instead of procaine as part of their regimen, and there were no reasons given for that, why that inappropriate exposure happened. And the corrective actions, again, included some type of personnel policy action and then following the subjects for their own safety. There were doses that were failed to be given. Again, the contributing factors not specified, a sign that the organization probably didn't understand, or at least didn't report their understanding to OHRP of why this event happened. And maybe if you don't understand why it went wrong, it's going to be really challenging to know how to prevent the next case from occurring. So those were some examples from the OHRP database of things that can go wrong in research, a very small sample that only rose to the level of being considered an unanticipated problem. Now remember, we did the study after both OHRP and an FDA issued their guidance on reporting unanticipated problems, and we waited about a year so that that guidance documents could seep into practice and could seep into people's SOPs. But even given that, out of the entire country, we only had 85 reports to that one organization. Well, so how about published cases? Well, let's look at that. Well, the Gusto trial was a trial conducted in the early 90s, also out of Duke University. As a matter of disclosure, I was the lead clinical pharmacist on that trial. Our pharmacy prepared all the drugs for Gusto for this worldwide study, 40-some thousand patients, multiple thousands of sites in dozens of countries across the world. Up to 13.5% of those patients had a documented and known medication error in the delivery of a thrombolytic to those patients. That's a lot of patients who got the wrong dose of a thrombolytic. When you look at this drug, it's a complex drug, and it's a high-risk medication with a narrow therapeutic window between safety and effectiveness. It's also a drug that had a very different administration schedule compared to any other drug of the day, with 3 different stages of administration. It's given in a complex environment of care, and it's a care environment that depends on the clinical practitioner and the clinical research team working hand in hand with well-coordinated, well-communicated roles and responsibilities. In the absence of any one of these things that have to go right, things can go wrong. And that's what happened with Gusto. The thing about Gusto that I'll make a comment about is, as a pharmacy in Gusto, we were entirely unaware of these reports in the field that were happening. The data were discovered in a post hoc analysis of Gusto. But had there been reporting systems that filter these reports and funnel those reports to pharmacy, to research officials and others running the trial, something could have been done sooner, I believe, to mitigate the risk to the next patient. So the reporting of these lagged years behind the identification of the problem, and then the surfacing of the problem lagged years behind the trial itself. You may remember the Tegenero study from the UK in 2006. This was the first in-human study of a monoclonal antibody. It was a rapid sequential design. They gave 6 healthy volunteers the drug. All 6 received the drug over 70 minutes. Within an hour and a half, they had a massive cytokine storm, which is not a good thing if you're a subject. It's a bad physiologic problem. And it resulted in multiple organ failure, requiring intensive care within 16 hours for these subjects. They were healthy when they went in. They survived with intensive support. Some had sequelae even at their 3-month follow-up visit. That was a known error. Not quite, right? We look back now with good lenses and say that was a problem. But what was happening about the time? In 2006, the New England Journal of Medicine said, you know what? This is wonderful science. We now have new insight into immune-mediated cytokine storms. But in 2009, there was a more informative editorial that said, you know what? The problem with that Tegenero incident was as much attributable to inappropriate design and conduct of the phase 1 study as it was to the pharmacology of the study drug. That author understood that there was a design failure that harmed those subjects. And we've learned from that. But look at the gap, 3 years between when it was blaming the drug, which is sort of an FDA drug safety science issue, and looking at the design of the trial, which is a science of safety issue that I talked about at the beginning. Two different kinds of concepts, very different outcomes. How about IV medications? Everybody has probably, unless you're in device studies, you're probably given IV medications at some point or another in a clinical trial. Well, Fell and his colleagues did a report because over a long period of time, their organization had multiple deviation reports from a single study where the IV drug was given at the incorrect dose, incorrect infusion rate, to the wrong patient, even the wrong drug was given. And those were reviewed by the IRB, and the IRB prompted the organization to do a root cause analysis, another good thing. Remember, they fell under the definition of deviations, but in reality, those were mistakes. Those were errors that occurred in clinical research. Following that root cause analysis, they showed improvement and a reduction of 60% in the rate of errors associated with IV meds. So the takeaway message is you can make things better. You can by understanding the root causes and addressing the corrective actions toward those root causes. How did they improve? Well, they looked at personnel. They said, you know what? We're only going to allow full-time nurses to administer study drugs at this organization. That became their organizational culture and policy. They also limited the administration of the drugs to a few units in the hospital so that you can control the exposure to patients under more carefully controlled conditions. And then they required 2 methods of patient identification to avoid you getting my medicine and vice versa. They looked at training and they said, you know what, we're going to train everybody. That wasn't novel. What was important was that it was competency-based training for the trials, so that assuring that the training was effective at improving knowledge and improving skills. Don't forget the skills part. It's not just the knowledge, it's also the skills. But the bigger issue that they did was to fix the systems that were designed with little flaws that allowed these mistakes to occur. They required electronic point-of-care documentation of drug administration. They required color coding on study drugs compared to regular medications. And you can see the other things. All of these strategies were done to improve the safe use of IV drugs following their root cause analysis. Each of these strategies had been known for decades before in clinical practice. And now, they had an aha moment, and now it's being done as part of clinical research. So again, another example, we can do that. We can move those practices over. Think about the labels on investigational drugs. Labeling of these drugs can be very high risk and are well documented to lead to errors. Brown and colleagues provided a nice picture in their journal of investigational product delivered to a research site with the first 3 products having nearly indistinguishable labeling, little tiny black and white font, all different drugs. The upper right-hand panel shows the drug and its placebo with almost imperceptible differences between the two. So easy to give the wrong medication, grab the wrong vial for administration. And the bottom right corner, I've experienced this myself, where product comes from the supply chain to the site, to the pharmacy, in big polyethylene bags with zip ties and a packing slip in the bag. That's it. That's how it arrives. How easy is that product to be mixed up on site? How much quality control has to happen at a local site level for that product to be distributed properly when it arrives under those kind of conditions? Why do we let that happen? Drug names themselves can be problematic, and the fact that, as I mentioned before, there are look-alike and sound-alike drugs. In clinical practice, those are called LASA drugs. Sometimes they're called salad drugs, sound-alike, look-alike. But nonetheless, they lead to mistakes because of confusion. One of the strategies to reduce risk is the use of camel case lettering or tall man lettering. You probably have heard this in clinical practice. So daunorubicin and doxorubicin, two potent chemotherapy drugs, they are more distinguishable by emphasizing part of the drug name in this camel case lettering, and vinblastine and vincristine, two medications that I have personally seen cause a subject's death because of their confusion. And vinblastine and vincristine can be further distinguished by camel case lettering. The FDA has references and guidances on drugs that should have this type of lettering. Other organizations also promote that as a safety design feature for drug labels. So we know that through the work of Dollinger and his colleagues that regulations increase the complexity of labeling, and that labeling is incredibly heterogeneous throughout the industry, with some good labels, some bad, and labels that change even within the conduct of a single study. The safety experience in clinical practice has been to use differences in color, font, and character size to improve and help distinguish between products to avoid confusion. If you are of a particular age in this room, and you remember when there were multiple strengths of insulin on the market, 40 units a mil, 80 units a mil, 100 units a mil, do you remember what the industry did to help you, the practitioner, distinguish between those three products to avoid killing somebody? They had different shaped vials, so that the tactile stimuli was another clue for those who overread the label. The tactile stimuli was another clue about which strength of insulin that was. A strategy adopted from the airline industry, where pilots have different shaped handles on the controls for their aircraft, so that even if they don't know where their hand is, they know that if it's on that particular handle, that's going to make the plane do a certain action. So, do we do that in research? Not so much. What did not affect error rates? The type of occupation you were in, pharmacy, nursing, physician, wasn't a factor in influencing the error rates. How experienced you were in clinical trials, nor your training. None of those were factors that affected error rates. So what does it mean? Well, labels are an independent risk factor for problems that can occur in clinical trials. The user experience and the training is not associated with error reduction. We need other strategies beyond user experience, basing it on the most experienced person in the team and training of everybody to fix that problem. We need to fix the flaws that are in the design of the system to reduce the risk for all users, regardless of their occupation or experience. So let's look at consent forms, because I mentioned in the objectives we were going to talk about drugs and consent forms and how those can reduce or contribute to risk in clinical trials. I'm going to talk about, in the amount of time, one little section of the consent form when a medication has the potential for drug interactions. It's very common in my years on the IRB to see the language, "Tell your study doctor about all of your medications." That's the simple language. Now, I don't want to limit this just to consent forms. So this could be in a patient diary, this could be on instruction cards, but regardless of the information the patient receives, it's "tell your study doctor about your medications." Take a look at a proposed revised text, because do patients think that their medical foods, their herbal products, their over-the-counters, and OTC drugs, do they think of those as medications? Many patients may not. So having this more specifically identified in the documentation improves the reliability of a good medication history, number one. And then number two, not only are you telling them to inform the study doctor about that at the beginning of the trial, but I've added for emphasis here, tell the study doctor before you take those drugs during the trial. In particular, when the study drug itself has the potential for interactions with a range of medications, herbal products, and medical foods. So these are simple strategies to begin to change our thinking and the way we look at otherwise normal text. But normalcy has to be challenged in this regard. So another example in consent forms. Take a moment. Just think about this for a second. Study drugs have many different names, right? Sometimes a study drug will have 2 or 3 different names in the investigator's brochure. Which of those various names should be on the consent form? Which ones should be used? All of them? Wow, okay. That's one thought. Great for the study team. But let's look at it another way. From a patient's point of view, now, okay, first off, if you are in the camp that believes patients don't read consent forms and are not important, just tune out for a minute, okay? Because nothing I say is going to change your mind. But if you are in the camp that believes that we can improve consent forms, and you know that we can, God, there's been enough talks about that today and elsewhere, then you might think that, well, what we put on the consent form ought to be what's on the product label the patient gets. Okay? The name on the label of the drug itself is the name that ought to be in the consent form, the diary, and information sheets that accompany patient materials. They need to match, because I can't expect my family members who are not in the medical health profession to know that this drug and this drug are synonyms. Even if you load all those up in a consent form, and I've seen consent forms that change the name throughout the document. They're not even written consistently. So let's use a name and then stick with it all the way to the dosage form and to the diary where that dose is documented. Well, I will give a shout out to one of my colleague competitors, the Institute of Safe Medication Practices. They've done a nice job. 2 references here for you to look at later on compiling some IND product-related concerns. And I'll just leave you with that because there's a lot of information in there to investigate and to learn from other reports. So learning comes at us from multiple perspectives. Some of you may know the Betsy Lehmann story. Just by show of hands, could I get a high five on how many know the Betsy Lehmann story? Wow, 2 or 3 people. Okay. This is a story that should be told to anyone entering clinical trials work, okay? Now, this is a story about a patient who was the healthcare reporter for the Boston Globe. Her story has been told in the lay media for 30 years since her death, about medical errors that happen in hospitals. In this case, a medication error that happened in hospitals. She died from a massive overdose of cyclophosphamide. What nobody talks about in that is that it was a part of a clinical trial. Nobody talks about the fact that she was enrolled in a clinical study and other subjects on her floor and being treated for breast cancer who were also harmed by the same mistake, the same overdose. That mistake wasn't discovered for 10 weeks after her death. They had no idea why she passed away unexpectedly. How was it discovered? The dose was too big to put into the electronic case report form. They tracked it back. Oh, we see what happens here. So Dana-Farber did amazing things after that. This was obviously a shocking aha moment for them. It was a high-profile event, and James Conway and his team conducted a root cause analysis. They wanted to make it right again, not just for this event, but for every patient going forward. So they conducted a root cause systems analysis to understand why the event happened. They focused on the systems, not the humans involved, so that again, what set up the human to fail in the design at the organizational and study level. And they made a commitment to learning and improving by developing procedures and instilling a culture of safety in research as well as in clinical practice at that organization. What did the RCA show? It showed lots of system failures, from systematic failures in training to problems with the way that the dose was prescribed, because physicians and nurses and pharmacists think about oncology dosing differently, different paradigms. And that paradigm difference wasn't understood prior to the RCA. And that paradigm difference said, docs think about total treatment doses. A nurse and a pharmacist think about how much I need to give for the next dose. And that was a factor in Ms. Lehmann and others on the floor that same day receiving much different doses. They had ineffective leadership, ineffective error reporting and error management systems. This is all out of the RCA. I have to admit that they published this in a private publication. It's never been in a peer-reviewed medical journal for others to see. But I think that this is an instructional case for you, your teams back home, to learn from about what can go wrong and how do we make it right again. What Dana-Farber did was two things. They made operational changes, but I believe more importantly, they made transformational changes. Operational changes we're really good at. Fixing the SOPs, requiring double checks, et cetera, et cetera. These changes that they had, mandatory supervision of doctors in training, double checking medications, and other operational changes. But they made transformational changes. They began a journey of saying safety cannot be compartmentalized into a single special team. Safety is the responsibility from the Board of Trustees, through the administration, through the PIs, down to the front line. Transformational change. Spread the word. Build a culture, the culture of being relentlessly vigilant for risk, error, and the potential for error that could result in harm. Improving the reporting systems. Having it possible for anyone on the research team to submit a report of concern that this practice that we're doing, this label, this SOP, could result in a problem to a patient, or it required some heroic measure to prevent the problem in a patient by doing things even the normal way, the required way. Transformational change. They looked at safe system design. They used technology as a forcing function so that they put a hard stop in the process, kind of like a timeout, if you're familiar with what goes on in the operative environment. There's always some timeout before a big procedure. Well, they used information technology to create that effectively forcing function that says, stop, let's make sure this is good. It's a hard stop. We can't bypass it until we can be assured that things are good. And recognize, again, that mistakes will happen, but we can develop systems to more quickly identify those, to mitigate the harm, and to develop systems to protect the patient and to protect the staff. And if you haven't heard of second victim programs, I would encourage you to look into that. It's a psychological welfare safety net for individuals involved in serious errors in practice or in research. So moving beyond the regulations for a moment, I will suggest to you that we should begin to think about clinical research as a component of the entire healthcare system and not separate from healthcare. Not clinical practice, not clinical research. They're highly integrated. Build that culture of safety, build that engaged leadership, and then view mistakes and errors as preventable harm and consider them as events of special interest. How do we improve the reporting and recognition? Well, have you ever thought about building into your protocols under the data and safety monitoring plan specific instructions for reporting medication errors. I am so pleased at the number of company protocols that I am beginning to see now that talk about all of the regular conventional research reporting of safety events, and now they have special language and special reporting for medication errors as part of that. That is beginning in the last 2 to 3 years, but more companies are taking this up. I've got slides here with some example language that I've anonymized and displayed to you from other protocols that are out there. So pharma companies are beginning to build this in, and IRBs should reinforce that and expect that. And site-based researchers should also build that into your own data and safety monitoring plans, collaborating with the organization's existing safety reporting program. So, when something does go wrong in a clinical trial, report it to the IRB and others per regulation, but why not also report it to the organization's safety reporting system? They have the expertise to do the RCAs, they have the expertise to investigate, and they see the bigger picture across all patients and all subjects at that organization. So it's a recommendation that as part of your policy, either at the IRB level or at the protocol level, that when we have an error related to medication, devices, or a procedure involving the patient, that should be reported to the organization's safety reporting program. And then focus our actions on the system failures and develop action plans that go beyond training and counseling, discipline, emails, and other temporal measures that we know are ineffective in preventing the next problem. So in summary, I hope that in leaving today's conference that you might be a little more vigilant and able to identify unsafe drug use practices and latent errors, those little hidden design flaws in the system that can contribute to a future problem, that we design our systems for safety based on what we already know works in clinical practice. And all of that will improve compliance with both ethical standards in the conduct of a study and the regulatory requirements as well. So I thank you for your attention. I hope that I've finished on time. And